Welcome to this final session of our meeting on the 100 years of genome. And I want to make a few logistical advertisements. First of all, there will be a switch in the order of talks, and Paolo Venez will talk before Takashi Goshi-Gori. And the second thing is that we are organizing a lunch after the end of the talk in the nearby restaurant La Limonaia, and I asked Giulia Trimani to pass around a piece of paper so that anybody who is interested can cross, so that we know how many seats we are going to reserve. And finally, as I told in Naples, most of the organization for this meeting on the part of the Academy has been followed and done by Annibale Montana. Annibale Montana couldn't make it in Naples, but he's here today. So I feel my duty to invite him to chair this session. And so for one time, I resign as president, and since they didn't prepare I did it. So Annibale, please, thank you.
Good morning, everybody. I'm very pleased to be here. Not so much for I have been compelled to run the session, but because I had a very good relationship with Giorgio Bernardi, and I feel my duty to make everybody know that he was my colleague at Roma Tre University for 10 years, and my colleague at the university the Academy delle Scienze Detta dei 40 for the last 2 years. He was the one who was most engaged in running this meeting together with Roberto da Novaro. He was actually the two nodes of the organization were Roberto da Novaro and Giorgio Bernardi. I only supervised for the section, practical section here in Rome. And as a supervisor for the practical session, I invited, I will, before inviting the first speaker, I wanted to thank you all who are here for remembering him. I ask your forgiveness in case I make some error, because I'm not an expert of genomics. And eventually I invited the first speaker to come and start his lecture. Please come ahead. Il primo speaker che è stata invertita la... Ok.
Thanks, Annibale. I'm very happy to be here, and because this represents the final result of 2 years of preparation, ideas promoted by Giorgio Bernardi. And just to recap a little bit what has been done in Naples, thanks to this joint venture between the Accademia delle Scienze and the Stazione Zoologica Anton Dohrn, last Monday, a fantastic series of presentations explored different fields of the genomics. And we started from the limits of life, even with Franco Salvatore, to the global ocean genome, to the evolutionary aspects, some insights into the human genome and the perspective related to also to the importance on the environmental conditions and epigenetics in setting the conditions for expression of genes and eventually changing the perspective of humanity and life and biodiversity with respect to many problems related to disease or just to the impact of the global change and whatever. So very excited about the first, presentations that we're also exploring and dig into the past of the origin of the genomic term, and span through the future, the present and future. For instance, Giacomo Bernardi exploring also the invasion of marine species crossing, and eventually the importance of genomic in understanding these aspects. We are here now to complete this journey and to also set up with some insights to set up the future. Some reporters were asking, the meaning of genomes. First of all, they were asking, how are we happy to be together in person, and then to start having, and I just replied, that's fantastic, because the COVID allowed us now to be together thanks to vaccination, but also to have a mixed, conference with people at home, safe, and following the. But the second issue is how important it is to celebrate a discipline that is young despite 100 years and is just starting again and providing huge perspectives for research in the field of life sciences. And I'm particularly happy that President Montana and President Concini are here, because I think that understanding the problem of genomics, in general, the genes, the importance of life sciences is a problem for everyone, because first of all, there are, we need to have multidisciplinary, interdisciplinary approaches. And we just, mentioned the importance of informatics, statistics, mathematics, modeling, whatever, also for life sciences and for the genomic studies. But because we all have to be aware of the main, issues of the future of humanity and the perspective of research that of relevance for all of us. So thanks again to the Accademia delle Scienze for allowing to the Stazione Zoologica to be here. This at the end was the objective of our connection, because we wanted to start a little bit, yes, providing an idea of the research done at Stazione Zoologica, but opening the scenario and inviting the Italian community and the global international community to share these important issues, and eventually to reevaluate the importance of the oceans in the perspective of the genomic studies. So thank you very much again, Livele. Thank you, Corrado, for everything. And that is nice. For me, time to give the floor to Massimo Milone and Max Mizzao, two fantastic, moderators that will accompany you in the second session. Thank you very much.
The first speaker is Massimo Milone. He will speak in Italian, I understand, but there will be translation immediately into English.
Buongiorno in questa splendida palazzina dell'Accademia dei 40. Proseguiamo il nostro forum aperto a Napoli alla stazione d'Oro. Sì.
Good morning, everybody. I'm very happy to be here. and especially of being hosted with you in this marvelous building, which is the seat of the National Academy of Sciences, the so-called Academy of the 40.
Ecco, ringrazio i presidenti dell'Accademia e della Stazione Dorno per l'invito. La mia è una testimonianza insieme al collega Max Spitzel. Il termine genoma, lo abbiamo già detto a Napoli, è stato coniato esattamente 100 anni fa, quando ancora forse si conoscevano appena i cromosomi come sede dei geni. Da allora le ricerche hanno rivoluzionato la nostra vita, basti pensare al nostro codice genetico svelato, alle possibilità di cura rispetto a molte malattie, alla biodiversità delle forme di vita sul pianeta. Abbiamo registrato a Napoli, alla Stazione Donna, delle interessantissime riflessioni scientifiche. Pensiamo al ruolo dell'ambiente, più che mai in questi mesi. Ieri sera proprio il telegiornale principale del servizio pubblico radiotelevisivo ci ha raccontato di un altro ghiacciaio, quello del Gran Sasso, che si sta sciogliendo.
Scusi. So I'm here with my colleague here, Max, and we're going to talk about the genome. We know that the genome has been discovered more, or probably little more than 100 years ago. And since then, many things have happened. And when it was discovered, actually, we virtually didn't yet know, or we just had known that it was the seeds of the genes. And so since then, a true revolution has taken place, which has really revolutionized our whole life. And for instance, with the Human Genome Project, which has discovered and decodified the genome, human genome code, and also opening up to possible treatments, as well as sharing information and insights into biodiversity and how to treat it, and actually enables 2 days ago, we have heard a lot about this in the Dawn Institute. And last night, during the TV news, we just heard that yet another glacier, the glacier of Gran Sasso, has started to melt down.
E cosa dire ovviamente rispetto a questa attualità scientifica dell'identificazione di microrganismi patogeni, più che mai in questo momento con il recente caso del coronavirus che interroga il mondo, insomma. Per dire cosa? Che seppur questa 2 giorni ad altissimo livello scientifico, insomma, ha registrato e registra oggi, insomma, giornata conclusiva, insomma, un confronto tra i massimi scienziati, la ricaduta sulla vita di tutti i giorni da parte del cittadino, cioè di tutti noi, è immensa. Più che mai in questo momento c'è esigenza di competenze. 1 o 2 giorni con questo livello scientifico, insomma, è una risposta di competenze, ma diventa anche una sorta, oserei dire, di servizio civile per un paese, per delle comunità che in Italia, e non solo in Italia ovviamente, il coronavirus non ha confini, si interroga su quello che saranno, insomma, le scelte di politica economica, di politica sociale e persino di relazioni interpersonali per il prossimo futuro. Un futuro che è già cominciato. Post pandemia è una parola molto abusata, siamo ancora in piena pandemia. Alla post-pandemia dovrà trovare delle basi solide di conoscenza per costruire una società diversa, una società con una economia ecosostenibile, con la possibilità di dare risposte di vita ai nuclei familiari, ai giovani, agli anziani, ai soggetti fragili. Queste scelte non sono lontane da un'aula come questa, non sono lontane dal lavoro meraviglioso che ricercatori in tutto il mondo fanno sui cambiamenti climatici, ad esempio.
Scusi.
Riflettere sui 100 anni del genoma significa anche riflettere sulla società del domani. Grazie.
So in the last 2 days, we have seen a very interesting and very fruitful sharing of ideas amongst the scientists and researchers. And now, probably more than ever, this is particularly useful and precious for us as all. And in terms, in particular, of the pathogenic organisms that have been identified, and so in order to spread the insights and information about this, especially now that we've seen with the coronavirus that there is such a very strong need for competence and expertise and knowledge in general. Is it really the name of the game, we might say, in the last, especially in these times. And this encounter and these meetings between scientists is important not only for scientific purposes, stricto sensu, but it is also very, very important for ordinary people, for the population, because it's the population in general which really is in great need of competence. And I would even say it's a sort of civic service, which is rendered by scientists and researchers, and especially during opportunities and occasions like this one, because this is a question that has an impact on all peoples, every single person, all communities, because now we know that the world is a whole community. There are no borders nearly every long. And this sheds a lot of light on the need of and the interest of peoples in general in knowing how the important decisions are arrived at. The important decisions in the field of economy, in sociological terms, and even, allow me to say, in terms of personal interrelationships. And this is for the future. This is for the future because we are not yet in the post-pandemic era, as someone thinks, because there is a lot still to happen, and because a lot to be done, especially to have an environmental friendly and sustainable environment, and to protect the most vulnerable strata of the population. And I have to say that this occasion we are going to have today, and we had in Naples, brings us closer to the rendering of this service, the civic service I was mentioning before. This is our work, and we absolutely have to we may do so by thinking back of the past, of what happened 100 years ago, and in celebrating the great discovery of 100 years ago.
Thank you. Non vorrei abusare del mio tempo, vorrei prima passare la parola al collega amico Max Spitzel, poi introdurre i nostri relatori.
Thank you very much, ladies and gentlemen, President and Massimo Milone, all friends here gathered. I'm very pleased. It's an opportunity for me just to mention a few thoughts. One is about Giorgio Bernardi, that we're so pleased to have Giacomo here today and yesterday and the day before. And I remember with Annibale and Roberto, the year we spent together in preparing, I was serving, obviously, our scientists and presidents. I was just an assistant, but I learned a lot, and I hope that these 2 days with also our friends from the major press and media like Massimo and Giovanni Caprara that will follow us in the public debate from the Corriere della Sera is also an opportunity to share this incredible international science experience with the younger generation and with policymakers, because at the end we are all united in one Earth and one planet, no? And so this is very important, I think, this opportunity. And the second thoughts is that we were talking last night, we were invited by the President De Concini at the House of the Aviator, and we were talking about that it's a big challenge for scientists to arrive to the ears and to the heart of the social community, you know. And this is not for granted. We were also talking about this with Julie Maxton. She's the lady of the Royal Society, executive director. And in England, we have incredible scientists capable to outreach and to share with the social community. So I think this is something we have to take in consideration. No? We are not in an ivory tower. Science, thanks also to the COVID, it's an experience where there is democracy. No, unfortunately, with the Earth virus like COVID and past COVID, we're all at the same level. No, so also for scientists, I'm not a scientist. I invite you to use all your scientific experience, heart and brain to share your knowledge that is so important, no, in this moment of the world, with also other stakeholders, no, with the family, with the class in university where you're a professor, with the social community, because we have to spread the word, no, around this. So just to share with you some thoughts about this so important international occasion. So please, I pass back the floor to Massimo to introduce the first talk and the first speaker. Thank you.
Grazie. Allora, la parola alla professoressa Cecilia Saccone, membro dell'Accademia Nazionale delle Scienze. Prego.
So I was very glad, very happy to be here, invited to this meeting, which celebrate 100 years of genomic research. And the reason are indeed very different. First, it was in honor of Giorgio Bernardi, who was my great friend, and I worked with him at the beginning of my scientific career, when for the discovery I will speak to you today, and the discovery of cytoplasmic genome. And well, I am coming from the University of Naples, so I know very well what as important has been the Stazione Zoologica di Napoli. And when Giorgio came as director, I was in the scientific committee. So this is another reason. But and so I am sorry not to be able to participate in Naples for some, really personal reason in this difficult period of pandemic of the COVID. But in any case, I would like to start my presentation. So during the lockdown, I wrote this booklet whose name is Il Codice dei Viventi, for a simple reason. During this time, I realized that nobody knew indeed one of the most important discoveries in the field of genomics, which is already 100 years old. And some years ago, I published in the EMBO, it's okay? Yes. Is in EMBO report, a paper calling the mitochondrial genome a neglected genome. So indeed, by the way, also according to what I have heard today, now we need real science. We need to spread the science. And so I wrote this small booklet, which only a little bit more than 100 pages, but contains particularly the discovery of the mitochondrial genome or cytoplasmic genome. And it was already, there are in the state and everywhere in the world, very, very high school. And I started in Naples with Franco Salvatore, and then who was the pupil of Cedrangolo, who was the pupil of Gaetano Quagliariello, who was the first biochemist. And so in that occasion, I met my husband, Ernesto Quagliariello, and he moved, and I was, the people offered me a place of assistant in the University of Naples. After I followed my husband to the University of Bari, and I lost the work. At that time, there was not possible to stay in a laboratory if this laboratory was directed by something until the seventh generation linked to people who should have the place. This was the reason, that the laboratory in Bari of Ernesto Quagliariello was studying mitochondria, and I too in Naples. But then, since in Bari, I had to move to another institute called at that time, not department as I started to be involved in that discipline, which is now more than 100 century old, more than half century old. So the discovery of mitochondrial genome. But this discovery, which is, I wanted to put emphasis on this point, has created, everywhere, big school is not known. So at the social and political level, and also at the level of university, I must say. So I worked a lot abroad, just to participate to this discovery, you know, since the beginning. And at that time, I remember Giorgio was also involved in this subject because he worked with yeast. So let me go on. So this booklet would like to emphasize what is the genetic material we are dealing on, and you know what is biology. And so in this small booklet I put as introduction what is the life, how to define the life. And it is difficult because it's a complex subject. But according to Rutherford, for example, Rutherford said that the only science is mathematics, then followed by its application in physics and, let me say, a little bit in chemistry. But he said that the biology was a stamp collection. That was the time when Rutherford died in 1937. But then when in biology, we had already in the century before the 20th century, a lot of important scientists who put the basis, what are the fundamental of the biology. And the fundamental of the biology, just to make life in a way simple. What is life? I using a very few words. Life is nothing except morphology, is energy, and is information. And so on. So if you think about the morphology, you can think to anatomy. But what about the energy? So the bacteria, you know that as a living matter, we have two types of cells, the prokaryote, which are shown in this, no. And what is this slide? Oh, no, it's okay. No, this is this is the prokaryotic cell, so cell which belong to bacteria. As you can see, they are called the prokaryotic from the Greek pro nucleus. They have a DNA, but this is not inside a nucleus. And then, well, we have questa qua. This is the prokaryotic cell. I think that everybody knows, very simple. There is the DNA, and the DNA was the information, of course, but nothing, a very simple structure. Then we have but we have the eukaryotic cell. According to the endosymbionting theory, is the endosymbiontic theory? Well, no, it's difficult. Endosymbiontic hypothesis, the eukaryotic cell, which possess some, you know, a nucleus and some organelle, has a structure which depends by according to the endosymbiotic hypothesis, to two bacteria, an archaeobacteria and an alpha proteobacteria. Archaeobacteria, which is a metanogen bacteria, and the alpha proteobacteria, which was a producer of hydrogen, with in the environment of an explosion of oxygen, they put together to form the endosymbiontic, the eukaryotic cell. According to, there is an example of the eukaryotic cell. No. Okay. There is the eukaryotic cell here. I can the eukaryotic cell in which there is a development of a membrane. To make the story short, this membrane also creates some organelles which are called mitochondria. Mitochondria, and is a very more sophisticated cell in which there are the nucleus, which has a great part of them inside the membrane. So there is a development of membrane in general, and in particular of this organelle. So this organelle, the mitochondria, so have a function, very important function, to give energy. Anyway, the word mitochondria derives from the Greek mythos, philo in Italian, and chondrion or organulo, organello, organelle. And this organelle has a structure which resembles in great evidence with bacteria. And so there is a symbiosis of two prokaryotic cell. So this prokaryotic cell, to make again the story short, created such organelle to produce energy which belong to the cell to which for all the eukaryotic cell. So more than 97% of energy comes from this organelle. And I studied in particular, there were a study on the function, and as I mentioned, and this was studied also from big school, but particularly for the oh no. For every type of animals. So the peculiar things of this organelle is that this one. No, for endosymbiontic hypothesis, and then this, okay. Okay, so the eukaryotic cell, it was allora, è questa che deve dare. Ah? Com'è? Ah? Which one is on the big? Questa. O questa? Questa. So I spoke about endosymbiontic. Questa è dietro, questa è avanti. No, io avanti, io devo andare avanti. Ok, this is the eukaryotic cell, and questa adesso nello schermo.
metà mi chiede passo io le slide, mi dica quando devo passare.
Allora, dopo di questo, so what was in this book are described the fundamental of biology, but I don't think I have time to show you this, and which are at the basis, like the evolution, the biodiversity, and in which mitochondria play a very, very important role. So let me go. So mitochondria, this organelle, which are in different number according to the function of the cell and the function of the animal. But the discovery was that they were, after the selection of the organelle, they still now maintain characteristic of bacterial cell as far as function, as far as biology, and what is the amount of information. So, in other words, we arrived to the discovery of the mitochondrial DNA. The discovery of mitochondrial DNA begins before the great discovery of the double helix. For example, for yeast, who, as you know, are not dependent, are aerobic and anaerobic, when we arrive at 1949, where Prometheus isolated some yeast mutants which were defective for cell respiration and hypothesized the presence of some non-Mendelian genetic character. So, in other words, this was some information was before. Okay, then we had some scientists demonstrated the mitochondria incorporate tricyclic thymidine, a marker of nucleoside. So other, like NAS and NAS, demonstrated the mitochondria contain DNA. And so during after the double helix discovery, there were a lot of people who hypothesized the presence of another genome inside the organelle. Then from 1965 to now 75, the studies on the every aspect of mitochondrial DNA in cell exploded, particularly in vertebrate cell. And it was observed by many groups, among this my group in Bari, the first synthesis of RNA. So a mother in the States, to quote Kroon and Simpson, demonstrated that they were able to synthesize the protein. So, and again, to make the story short, with every eukaryotic cell which possess which respire, so uses the oxygen to produce energy as mitochondria. And these mitochondria are their own their own genetic system. And this genetic system is very similar to the genetic system of prokaryotes. And so, for example, I worked on the sequencing of the mitochondrial DNA from rat, and which is, as you know, has the same structure. So, what is the size of this circle? And usually it's a circle, but not always. There are a lot of the different size of mitochondrial DNA, but in vertebrate, it codes for gene for RNA, gene ribosomal RNA, 2 species, one genes for transfer RNA of less than those in number as those used in extra in the cytoplasm. In the nuclear cytoplasmic synthesis of protein. So they have their own synthesis of protein, which is, of course, sensible to mitochondria. What is they code only for 13 amino acids. According always to the Endosymbionting Theory, the alpha protobacterium give the information to the main, and so now we have this enormous amount of information in the nucleus, and a lot of information which is what is the size of this size of this genome is only between in the vertebrate 15 and 17,000. You know, it is half of the COVID as number of monomers. Of course, this is DNA, so double helix everywhere. And then it was in that time they discovered the genome in what was going on the plants. Also plants have a third genome. So they have the nuclear genome, the mitochondrial genome, the plastid genome. The plastid genome has just the gene for the photosynthesis action, and so they are responsible of life. So, this was we were at that time. I give a date of the reaching the illumination, the moon, the moon, and it was 1969. Everybody could know that we possess a mitochondrial genome. But this is not the case, so I still to see how neglect this is this genome. So to summarize, the animal possess 2 genomes, one in the nucleus, another one in the mitochondria, and the plant 3 genomes, 3 types of information. And of course, those of this tiny genome for both for plastid and for mitochondrial from mitochondria, they give all the energy we need. Okay, so but what is this another important discovery? The next one, this is the maternal inheritance of mitochondrial DNA. So the information which is inherited in the nucleus is half of the father, half of the mother. okay, that we have in the mitochondria is only inherited by mitochondrial DNA. You can see in this slide, for example, a couple, red and blue, which have both male and female with the same genome of the mother. And then they marry, and always they have, so the inheritance of mitochondrial DNA is matroclinic. So there are some exceptions. Recently I read some paper in which was found also the intervention of the father as far as the content of DNA of the father. There was some presence, but are still a very exception to the role. The role in the inheritance is always given by the mother. So. And we arrive at the discovery of mitochondrial genome opens to some important implication and also application. And as far as the implication, you can say, for example, this is a type of genome which is, which is subject to the action of many antibiotics because it's of the type of the bacterial. But there is a problem. So the mitochondria, I show you very quickly, as a double membrane. One the outside the membrane and the inside the membrane, so as should be permitted. And for example, several mitochondrial several are impermeable to the antibiotics, but the majority, particularly with long-term therapy with antibiotics, demonstrate clearly that there is a damage to the full function of mitochondria, and so on. So now particularly important because the use without any integrated from the doctor in medicine for everything of many mitochondria, they causes some have a great effect. Also because the prokaryote have become antibiotic resistant. And so this is one of, dangerous, not depending on the mitochondria. But, for example, there are many other difference which make we become an idea what is the life, what is our life. Our life is a system, so it is not which it is done by very interaction. And so what it is really meaning is this interaction. So, the complexity of life is due to this particular case. And then I have, there are a lot of mitochondrial disease. In this book you can find a little bit more, but I try to compress all this idea in this very small book that for the first time I wrote not in English but in Italian, but which is in every library. So what was the advantage to have such not simple meaning, which is why to have two, the reason is very simple, is because the information, it is not important to have big information, because there are prokaryotes which have a lot of information, and they don't evolve. So the evolution, which one of, and the fact that the biodiversity is all depending by energy. And for example, the evolution could be done by information only, because there is a lot of a need of a lot of energy to transform this information in gene. So all the world of animal or world of the plant are due to the energy of this small organelle. This small organelle, it is clear. So which open the possibility to convert through protein synthesis to a small number of genes, only energy in other characters, and so to create the system as should be now. But this, of course, there are a lot of diseases depending on the mitochondrial genome. But as I mentioned, mitochondrial genome in our developed organism has only a few proteins which are you know, of the primitive prokaryote. All the others, all the information be transferred to the nucleus. So the nucleus can manage all, because of this situation, a lot of energy to create a new character, a new type of life. Okay, and among these, so the mitochondria are, as energy, are, you know, very, very important for our actual reason. And it's but there are also some important applications, and this is the maternity, assisted maternity. So we come to the definition of sex. Sex. What is the meaning of sex? Sex is sectum, from Latin separated. There are two genders, and you know, and one possessing, they both possess mitochondria, but the gender, the masculine gamete, they also as the mitochondria, and this help to reach the ovocyte. So it's a mobile, but which can reach the other, almost all gamete of the female sex, which are, as in our case, can be the cycle during oogenesis and embryonal development, which is very, very important. And so you can see that the primitive germ line is very as the stem cell are very limited number of molecules of mitochondrial DNA. Then, as you can see here, the ovocyte, which is mature for the fecundation, they have to possess not less than 1,500 molecules of mitochondrial DNA. And they are, what is the reason? When they are fused together, the plasma and the gamete, the ovocyte, they give rise to the embryon. And the embryon, whereas during the maturation there is a lot of duplication of mitochondrial DNA molecule, after this stops at the level of after fecundation, and then goes after only after the several, after the implantation in the uterus until the blastocyte. Then resume the protein, the synthesis of mitochondrial DNA. So this is very, very important. And I want to show, excuse me, is it time? 5 minuti, sì. Allora, then, so you can see here, I hope clearly, that so this is the way we reproduct. Okay? Then there are in this book other interesting application, like in the cancer side. And you will discover, because I have no time to explain you, that the mitochondrial DNA enter not only in the production of a new organism, but only for the life of the, but also for the dead, so the apoptosis, because is responsible of the liberation of cytochrome C, one of the protein which are important for and just to make my talk, mitochondria is a common factor for vital activity. So you can see, since it gives energy, it for vital activity gives for all the metabolism, for the reproduction, and for there is the big theory of aging, because aging, what is the mitochondria, like that produces variant, like the COVID, for example, and we become aged. So we become aged, and then also there is in what is also important for this aspect, to be to work in line and not in contrast with what does in the nucleus. So this is and again we become to the system biology. So for in case of the COVID, I discovered not only, even before, that in social and political aspect, very few of people, they know that this is the reality. if we have not, if we don't care of what is happening in the mitochondria, we cannot have life. And so I wonder whether in these conditions, for example, for everything which concern even the vaccine, even the reaction of the vaccine, why it is not taken into account what is written in this small booklet. Thank you very much.
Thank you. Il professor Paolo Vinaes dell'Imperial College di Londra.
Thank you very much for inviting me to this very interesting meeting. Can you hear? Yeah. The theme I've been assigned is gene-environment interactions, and I will tackle it from the point of view of non-communicable diseases, with a particular focus on epigenetics, which is the field I work in. So what are non-communicable diseases? Well, we probably all know that by non-communicable diseases, we mean cancer, cardiovascular disease, diabetes, hypertension, and so on. However, this is a kind of imperfect category because it is defined as opposed to infectious diseases, which is not great. And in fact, there was a very influential paper in the Lancet a few years ago that proposed to reframe them. They said that they are a kind of ragtag group of leftovers that do not satisfy Koch's postulates. So they described non-communicable diseases, suggesting to call them socially transmitted diseases, with a number of attributes, including chronicity, global burden, preventable nature, having common proximal risk factors like cholesterol, blood pressure, glucose, obesity, common behavioral risk factors, smoking, alcohol, diet, and others, and so-called distal risk factors, particularly economic, social, and environmental. And they all involve common issues of inequality and injustice. So this is particularly relevant today because we are observing increasing inequalities in the world, and this also concerns non-communicable diseases. Now, I would like to start with a few challenges in the epidemiology of NCD or socially transmitted diseases with the example of cancer. And then I will come to gene-environment interactions. So first of all, I would say decades of research on the causes of cancer, probably starting with the seminal book by Richard Doll and Richard Pito in 1981, suggests that only a small proportion of such diseases is due to genes, in the sense of highly penetrant genes. Though there is an important component of interaction between low penetrant genes and the environment. What we know now is that we are able to explain about half of cancers with risk factors, which are both behavioral and environmental. So we don't know almost anything about the remaining half of the cancers. But in the meantime, we know that not more than 5 to 10% of cancers and other NCDs are related to highly penetrant genes. And how do we know that? From much research which has been done in the last decades, but also from very simple observations. For example, cancer changes in time very rapidly. These are trends in the incidence of cancer in selected countries in recent years, with increasing trends and then a tendency to a plateau in some of the countries. recently. This is perhaps the most well-known example of the fact that cancers are essentially due to environmental or behavioral causes. It is the crossing of the curves for breast cancer and cervical cancer in low-income countries or countries that are undergoing a rapid development, in this case, India and Thailand. And this shows that cervical cancer is decreasing rapidly thanks to increased hygiene and screening, whereas breast cancer is increasing rapidly for a number of reasons, which include also reproductive habits. Then we have seen a very powerful phenomenon, which was the increase in incidence and mortality from lung cancer in the last century, which was clearly due to cigarettes. And in fact, you can plot the sales of cigarettes against the incidence of lung cancer, and you have a very clear overlap, though you have to consider the sales of cigarettes 20 years before, because there is a latency period before cancer develops. So this is a temporal dimension, changes in time. And then we have a geographic dimension, that is a cancer varies by geographic area very much, so that we can say that there are cancers which are typical of certain regions, like stomach cancer has been typical of Japan for a long time. Skin cancer of Australia due to UV radiation, or cervical cancer in Brazil and other areas due to HPV and sexual habits, and so on. So this tells us that once again, it is not genes, or at least highly penetrant mutations that explain cancer. In terms of mechanisms, we have to be aware that the onset of cancer is due to a sequence of different changes, mainly in DNA, but not only in DNA, which include mutations, but they also include epigenetic changes. And in general, people have described the onset of cancer as a kind of Darwinian process at the level of the single cell. That is, you have mutagens, which are very well known, but also selectogens, that is, exposures that select certain mutations. So many mutations are neutral, some mutations may be beneficial to the cell. Other mutations induce the type of cell proliferation that characterizes cancer, and they are selected by selectogens, that is, environments that are lead to the selection of specific mutations. So this is called branching evolution, and it is the current explanation of the mechanism of cancer. Many mutations are related to environmental exposures. There are certainly spontaneous mutations, but the more we look for environmentally related mutations, the more we find. This is a paper we have published a few years ago with Mike Stratton, and it shows the fingerprint, so to say, of mutational fingerprint of tobacco smoke. As you know, tobacco smoking is a very powerful carcinogenic habit. About 90% of lung cancers are explained by tobacco smoke. And what we observed in this particular case was a signature of tobacco smoke in lung cells of people with lung cancer who were smokers. And what was more interesting was that, well, first of all, this signature was different from the signature found in other types of organs. For example, bladder cancer has a different signature which indicates that aromatic amines are involved. Here we found that the signature associated with lung cancer was the same found in cell cultures treated with benzopyrene, which is a polycyclic aromatic hydrocarbon which is found in tobacco smoke, but also in the exhaust of cars. Car and diesel exhaust. In fact, PAHs and benzopyrene are a group of carcinogens that are extremely widespread in the environment. Cooking of organic material leads burning organic material leads to PAHs. So we found the same signature in lung cancers biopsies and in cells treated in vitro with PAHs. And then I would like to move to epigenetics. I said essentially two things about genetics and genomics. One is that inherited mutations or particularly highly penetrant mutations or variants explain a small proportion of NCDs, though there is a share of gene-environment interactions. And the second thing was that we shouldn't forget about somatic mutations, not inherited mutations, which are induced by environmental carcinogens or mutagens. Well, and then the third message was that we shouldn't forget about some of the Darwinian principles, that is, in addition to mutations, you need selection. So from the epidemiological point of view, we also study selectogens, that is, situations or environments that facilitate the selection of certain mutations and lead to cancer. Now, what about epigenetics? There has been an explosion of epigenetic research in the last decades. And you can look at epigenetics from many different perspectives. Since I'm an epidemiologist, the first perspective I was interested in has been looking for good markers and long-term markers of changes at the DNA level. And we have been using essentially DNA methylation, which is one of the forms of epigenetics. So DNA methylation is involved in gene transcription, and it is interesting from epidemiological point of view for at least two reasons. One is that it is reversible. Mutations tend to be fixed into the genome unless they are repaired, whereas epigenetic changes are reversible. So they come and go with exceptions. The second characteristic of epigenetic changes is that there is clear evidence that they are related to environmental exposures. This was already suggested by Waddington in the 40s to explain embryonic development as the interaction between genes and the intrauterine environment. Think of the importance of folate, for example, and how folate interferes with epigenetic phenomena. So I alluded to reversibility of epigenetic changes. Once again, this is an example concerning smoking. We studied smokers and we looked at epigenetic changes in the course of time. What we observed was that first, that there were a lot of epigenetic methylation changes in smokers. A large number of genes were affected. This is done with the Illumina technology, which is a epigenome-wide, which allows epigenome-wide association studies. So it allows to study nowadays 850,000 CPGs. And so for many genes, there were changes that were mainly based on hypomethylation, so a reduction of methylation. Well, I remind you that methylation of a certain CPG in certain regions of DNA tends to repress transcription, whereas demethylation leads to increased transcription. So hypomethylation essentially means that you have increased expression of a number of genes in smokers, and that leads to the changes we know in smokers. But there was also a reversibility of those changes. So after sometimes months, years, you saw methylation levels of those CPGs that were usually hypomethylated, but some were hypermethylated, went back to normal, to the situation before smoking. However, there was a kind of a series of genes that are indicated at the bottom in red, whose methylation didn't go back to normal. They stayed hypomethylated for a very long time, something like 30 years, after quitting smoking. Meaning that, well, if you like, there is a good message and a bad message. The good message is that methylation is mostly reversible, meaning that the effects of the environment can go back, can be reverted. And we know that people who quit smoking go back to the risk of never smokers, unfortunately, after many years. It takes at least 10 years to go back to the risk of never smokers, even more. For lung cancer, for myocardial infarction, it takes much less. It takes a few years. The bad message is that, however, for certain genes, for example, we found very strong associations with a gene which is called AHRR, which is related to the AH receptor. For those of you who have knowledge of biochemistry, you may know that the AH receptor is quite important in the communication between the nucleus and the cell in general and the external environment. So dioxin, for example, acts through the AH receptor. So for those genes like AHRR, there was no reversibility. So the observations are mixed, and you can keep a certain degree of risk of disease after environmental exposure, which doesn't go away, including certain mutations, which is a good, which has an interesting implication from the public health point of view. That is, you shouldn't smoke rather than smoking and then quitting, because quitting is not sufficient, so to say, to lead you back to the original risk or to a risk zero. Now, I would like to say something about our work on life course epidemiology, and particularly social epidemiology and social epigenomics. Well, I allude a little bit to sociology because first, the impact of social circumstances is coming in a powerful way to our attention in these years. We are now understanding that social inequalities explain a large proportion of diseases, and they are growing in many countries. They are very good examples in the United States, for example. So we are compelled, in a way, as biomedical researchers, also to include social disadvantages and circumstances into our research. And we shouldn't be naive in considering the social component. So, for example, I refer to a French sociologist, Pierre Bourdieu, who has written very influential papers and books on this topic. And he introduced the three famous capitals, economic, social, and cultural capital. So what a person is at a certain point in time is the accumulation of these three types of capital. You may be rich or poor, economic capital. You may have a lot of social relationships, which increases your likelihood of having success in life, and also the cultural capital. Now, what we have proposed in a consortium that I led, funded by the European Commission, was to add also the biological capital. Because at birth, you have inherited certain genes, and that's already a kind of biological capital. But then the environment induces many changes, for example, epigenetic changes that increase or decrease your biological capital. So what can happen is that a person at the age of 50, given a certain economic, social and cultural capital, may be in good health or bad health because of also of the accumulation of biological capital. And so we propose to put together these two concepts, which are biology, zoe, and biography, bios, to found a new approach, which is a life course approach to health. And this is a description of how things happen if you follow people from birth for many years. These are very famous Finnish cohorts led by my colleague Margherita Järvelin at Imperial College. What they show is that if you distinguish less advantaged and more advantaged people, in fact, boys and girls, in blue you see the less disadvantaged, so the richer, so to say, and in red you see the poorer. Well, you have differences increasing already at the start of life and sometimes diverging in the course of life. The poorer tend to eat less fruit and vegetables, they do less physical activity, they smoke earlier, and at a certain point in time, around the age of 25 or 30, you start observing also a divergence in terms of BMI, obesity, cholesterol, triglycerides. And the end of observation was at the age of 55, and there was a clear difference in terms of diabetes and cardiovascular disease in these people. So this stresses the importance of life course approach. In other words, things happen to start already in early years of life in terms of bad habits, exposures to environmental factors, and poor health and poor aging. Quanto tempo ancora?
Un minuto.
Un minuto. Then, well, I go directly to this. Very quickly, you can investigate what I said through biological markers, including epigenetic markers. So we have developed the so-called biological clocks, which are based on epigenetic marks, methylation of CPGs. You can use several different clocks which explore different functions of the cell and are associated with different outcomes like mortality. Well, the first clock was developed by Horvath in California. Now we have many more. They work very well because they describe the life course trajectory of people. They are associated with behavioral risk factors, and they predict mortality. So this is the first study we did where we looked at this biologic clock, also called age acceleration, based on DNA methylation according to socioeconomic position. And just look at the top right quadrant. You see age acceleration in high social class, then intermediate, and then low social class. Low social class is the black square, and there is clearly an increase in age acceleration depending on social class after adjusting for behavioral risk factors, for smoking, obesity, hypertension, diabetes. And then we repeated this in 16 different populations for about, I think, 18,000 people. And we found the same. Essentially, we used 4 different clocks, and the results were exactly the same. Now, I know that there is no time, and I will just say one word about the exposome, which is this new approach we use to investigate gene-environment interactions. The word has been introduced because people realize that exposures, environmental exposures, are really important in explaining disease. And so we have to invest resources and efforts into the study of environmental exposures. And this is an example of how you can improve the investigation of environmental exposures. This is metabolomics applied to food intake. So these are metabolites in the urine. You see that 14,000 features were identified with mass spectrometry. So it is really a kind of screening of all the metabolites you have in urine. And you see that in the heat map, each type of food is associated with a particular spectrum, like coffee, wine, orange, chocolate. All of these spectra are different, meaning that each type of food leaves its own metabolomic signature, which is really an improvement in methodology of epidemiology. I stop there. Thank you.
Thank you very much, Professor, for your lessons. And also, if I may add a little anecdote, now that we're passing through the last talk, and before you leave, is that I don't know if you read about this book 10 years ago called titled "If Nothing Matters" of Jonathan Safran-Foer. And the book is just a word, but it's a synthesis for our kids and daughters, also for us as fathers and mothers and grandfathers and grandmothers, about how important is food. You just mentioned it, and also what we eat, you know. And the book is about this father. He just had a baby, Jonathan Fraser, a great writer, that wants to explain why it's important to eat less food, less meat, and especially less intensive farming meat. And so he goes for 3 years around the United States, risking his life, because when he was going near this big farming, high intensive, they might shoot him. In America, it's for private property. And this is the story I invite you to read, because it gives a very synthesis of many important issues you are talking about behavioral aspects, environmental, and capital related to biological impact. So, and also this gives, then after the last talk of Professor Gojobori with my friend and colleague Giovanni, could be one of the themes of the public debate, the relation of what we eat, what we are, and how we live. So thank you so much. So please, now the floor is for Professor Takashi Gojobori. And as we said in Naples, Giorgio Bernardi was talking a lot about with President Danovaro and President Montana about having in Italy Gojobori. So I'm really pleased that he's here for us. Thank you so much, Professor.
Okay. Thank you so much, Dr. Patel. Very nice introduction. And also, I would like to thank President of Academy, Professor Consini, and President of Stazione Zoologica, Professor Donald. And here, could you give me the my slides? Okay. Thank you so much, Roberto. And let me speak again. And first, I really appreciate Professor Consini, President of Academy, and also President of Stazione Zoologica, Roberto. Really, thank you so much. Today, I would like to talk on what is the Genomic Information Society. I named the Genomic Information Society, which is really, we like to see the future based on genomic information. The mission is, first, really we have to consider genomic diversity and also big data. Then how we can see genome information, maybe oriented society in precisely. Then, just like 2 days ago, we learned from Dr. Christine Grover about how Hans Winkler thought what the genome is. And if Hans Winkler can be called as the inventor of the term genome, then I would call Professor Giorgio Bernardi is the leader or pioneer of structural evolutionary genomics. And also he worked until he passed away. Then really he was a man of structural and evolutionary genomics research. And therefore, I think his career would be a kind of a chronological time indicator when we see retrospectively the development of genomics. And as we know that 2 days ago, Dr. Maria Constantini showed how he worked on structural and evolutionary genomics from academic contents. But here I would like to show some career of his development. Then I think after showing briefly his career, then I would like to look back the development of genomics from my own contribution. So as you see that he obtained MD in the 1952 or 53. Then also already he worked on physics and physical biochemistry, and he was doctorate at the I'm sorry, I cannot pronounce French, But anyway, science, physicals. So, and then maybe it goes next. Say he was a medical officer in the Italian Air Force. And also he was telling me he was the pilot. And even he flew under the bridge in Venice, something like that. So he was very good at operating fighter. And he was assistant department of biochemistry, like in 1956. And he became a research fellow and a postdoc in Canada. Then he went back to Europe in France and CNRS. And also he was in charged the researchers and visiting scientists in US, and also director of research CNRS around 1973. Then, as you see, he became scientific council of Statione Zoologica in 1975. Then I think co-director of Institute Jack Mono. Then he became president of Statione Zoologica 1998 to 2007. Particularly his great contribution from my perspective to Statione Zoologica, he introduced molecular evolution into the research in Statione Zoologica. Then he wrote a book called Structural and Evolutionary Genomics. He published this book in 2004. But immediately next year, the book was reprinted. So we can see how this popular became so important to many researchers. Then he was President Emeritus from 2007, and he became a Vice President of IUBS. Then finally, he became a President of International Union of Biological Sciences, IUBS. And that was 2012. Then I think 2016, actually he visited the KAUST as a visiting professor. And just before that, I think I have to mention that he became a visiting professor at the National Institute of Genetics in Japan. So we really had a very nice time at that time. So this is a kind of his career. Then just keep in mind roughly his chronological order. Then I would like to go to development of genomics. Of course, as you know, like PacBio, these are becoming long strand sequencing. It was possible. Then nanopore technology invented USB sequencing, maybe for bacteria, just only 2 hours for sequencing. And for maybe viruses such as SARS-CoV-2, maybe only 1 hour. If the precision becomes much more precise, then on-site, instead of PCR test, but the on-site sequencing of SARS-CoV-2 would become possible, which means immediately we can know which variant you got infected. And also next generation sequencer, because next generation. So we use next generation sequencer as inclusively. But as you see, those sequencing is not only genomic sequencing, metagenomics, also gene expression, and messenger RNAs, transcriptomics, and chipseq, protein DNA interaction, even the PPI, protein-protein interactions, and epigenomics, just we had the lecture. So then a little bit from my own contribution, just parallelly looking at Giorgio Bernardi's career. Actually, my team, 7 people from my lab contributed the Human Genome Project. The 7 people, including myself, were the co-author of about 400 co-authors in this Nature paper. And I have been director of DNA Data Bank of Japan in collaboration with EBI, NCBI at the GenBank at NIH for 25 years. So therefore, we collected all genomic sequence data from Japanese contribution. We submitted this team. And around 2002, I contributed the genome sequencing of rice. And it has been so much competitive, but in 2005, we successfully coordinated the International Genome Sequencing Consortium. Then we sequenced the whole rice genome. Then around the same time, I utilized all supercomputer at that time over Japan Island, about 5 supercomputer center connecting CPUs. Then we made a pairwise comparison of the then available like 150 complete microbial genomes. Then we estimated what percentage of introgression from other bacteria, which is very important because mostly those introgression will be pathogenic. Therefore, in this case, we used Bayesian method. Currently, you might call machine learning. Then Dr. Hayashizaki at Riken, I initiated the kind of hackathon and also annotation jamboree for gene cDNAs, which is now called Mouse Full-Length cDNA Project, we call Phantom. Then we published quite a number of Nature and Science papers. Also, 2009, we published one paper in Science, particularly for Asian population genomics, how SNPs are distributed over Asian regions, which is very important to trace the origin of human migration on the Earth. Then 2010, we found the non-retroviral RNA viruses element, which is embedded in the human genomes. Actually, this is a kind of initial sign how much we have retrovirus or viral remains in a human genome. I would say more than 50% of human genome would consist of viral remains. Since we're interested in evolution of the nervous system, how evolution started in a nervous system, we worked on Hydra. This is very tiny primitive organisms, Cnidarian. Then we sequenced the whole genome. This is tuna, sushi. Because I'm Japanese, we sequenced the whole genome, Pacific bluefin tuna. And that is very interesting, like how it swims so speedy, and also like how they are going to consume energy. That is really important. More importantly, we discovered their blue light sensoring opsin genes are a little bit different from others. Therefore, they can see the sight, even pelagistic ocean so quickly. If we sequence tuna, why not nori? So we sequenced also sushi nori. This is a red algae. The red algae is very important because very, very old organisms. Therefore, if you are interested in origin of life, certainly this sequence would be very important. But technically, it was so difficult because mostly biosynthesis, so a lot of bacteria together. So therefore, we cannot distinguish this is really a red algae's genome or bacterial genomes. So we successfully grow in a non-bacterial condition. So we found that this genome is so small, and how this nori red algae grows is totally depending on which bacteria has inside of nori. So then those are kind of a timeline of the genomics, and particularly please remember Giorgio's career too. Then omics has been proposed. As you see, what is omics? That the genomics and the transcriptomics, ribonomics, proteomics, everything, the last omics. So therefore, without making any working hypothesis, we can generate the data, big data. This is called omics. So some people criticize this approach, particularly Sidonie Brenner. Dr. Sidonie Brenner criticized low input, high throughput, no output. So which means you can generate a huge data, but no intellectual output. However, this criticism should be reconsidered because from big data, we are extracting so important information. Not only such useful information, it can be utilized for aquaculture or agriculture, even environmental conservation, or even new energy and new foods. So certainly such a big data called omics is giving us the bright future. So as I said, the Sidonie Brenner, low input, high throughput, and no output. But so at that time when next generation sequencer was booming, at that time we needed a huge sequencing center. But now it's completely changed. We don't need a sequencing center. Maybe you can sequence by yourself, or you can ask the company. So therefore, sequencing effort itself, it's not really a big issue. What's the issue? What kind of biosamples you can take and how you can analyze the data. So Statione Zoologica has a tremendous advantage of those kind of features. And also from a human medical sciences, particularly in genomics, we thought that the fundamental institutions or fundamental knowledge can be transferred from hospitals or clinical stations. But this expectation was wrong. I think now hospital is generating the sequencing such as COVID-19, SARS-CoV-2. So huge data coming. The issue is how those data can be interpreted. Particularly real-time monitoring will be important. So therefore, how you have construction of sequencing capacity would be crucial, particularly to deal with such a pandemic situation. Moreover, sequence information would give just tiny knowledge. But if we can integrate other knowledge, such as various databases into, we call annotation to the database, the information will become much more valuable. In order to do so, I think we have to have an integrated database. So integration of database does not mean to include everything. I think once you have set up a given set point, vision point, then you can pick up those data to make everything consistent. This is integration. Particularly if we are interested in an evolution point of view, this integration will become much easier, much consistent, or much comprehensive. So 2 days ago, I also showed this is a message that we published a paper in Nature almost 12 years ago. But still, we shouldn't forget experimental validation is essential. Without experimental validation, only prediction is too dangerous. So therefore, curation annotation would be very important, particularly for constructing database. And from database, as I said, validation can be done. And once we have such curated database construction, we can utilize AIs, or we can share the data, not only clouds such as Amazon, but maybe we can utilize blockchain system, which can provide very good security features too. So from here, since 1990, about 2000, we conducted, as I said, the phantom, functional annotation of a mouse genome. Also, I initiated around 2000 human invitational transcriptomics project, which means we shared all cDNA transcript, including NIH in US and Europe, and we conducted annotation jamboree. And then we worked on genome network project, cell innovation project, and the structural life science project, which means all the data should be integrated to find out what would be the best way to analyze the data to extract the knowledge from genomic information. So we published those papers in Nature PLOS Biology. At that time, first issue of PLOS Biology, then Science. And currently, the number of human genes are assumed to be about 23,000 to 24,000. But please remember, all small coding protein regions are completely neglected. Particularly, the number of amino acid is less than 80, completely neglected. Therefore, if you include those smaller genes, then the number of human genes will become 38,000, even 40,000. So therefore, our human genes are still missing a lot. So in a cell innovation project, which is toward single cell analysis, we developed a pipeline. So bioinformatics tools called pipeline would be essential. Just you can push the button, then a certain analysis can be done, such as comparative gene expressions, and then even epigenomic identification of modified nucleotide. So we developed more than 300 pipelines. So therefore, these are very much important. More importantly, I think particularly nowadays, monitoring would be crucial. Like when you look at automatic driving, you don't have to drive by yourself. Just you get on the car, then the car would be automatically drove for you. And why that is important? Because the car itself can be a sensor, not only looking at the other cars, but the car measures the CO2 conditions around itself. Then the car each car can make communications. Even once you sit down the driver's seat, and your blood pressures, your of course weight, and those health conditions can be measured, and those data should be transferred to data central analyzing center. So, but anyway, then as I showed that we worked on marine metagenomics, and already I told quite much about what we have done. Just I like to skip. So therefore, when we conducted marine metagenomics, we sampled time series, like every 2 weeks at a given site, given depth. We worked on how those metagenomic analysts can see microbial community at a given site, a given time. In order to do so, we developed automatic seawater sampling robot. And even on this V, I think we can set up nanopore sequencing. So therefore, every, say, 2 weeks, the automatically seawater will be sampled. Then the sequencing will be done immediately. Then the data, it goes to the center. So therefore, if we can distribute those sensors along the land, we can measure how microbial community is changing, because it is important not only fishery, but also environmental monitoring too. And I moved to Saudi Arabia. We worked on the Red Sea, and then we did the same thing in the Red Sea. Then we found that completely different changes taking place. The microbial community in Japan has clear 4-season cycles. Every year, almost the same cycles. But the Red Sea, no pattern, always random. Also, we constructed the pipeline. This is not bioinformatics pipeline. This is actually from seawater sampling, then the sequencing, annotations, then we made single cell screening, then gene modification can be done, then we can have so-called value triangles or value chain. So if you are interested in a drug development, at the top, the unit quantity is so high, but still the target quantity is very small. But if you are interested in animal feeds, per unit cost is quite medium, but you can get large amount of entity. And if you can have an energy source, new energy, then unit price should be small, but a huge amount of quantity will be made. So anyway, we designed the microfluidic droplet screening, and if you can insert unknown genomic fragment from a huge amount of metagenomic data, each fragment can be inserted in an E. coli or Bacillus subtilis. Then 3,000 droplet can be generated. Then at the end, there is a sensor. If it contains the lipid encoding genes, it changes the color. So we can select only lipase, such as this. Why we worked on lipase? It's a huge market of detergent. The same way, this micro droplet screening will be so useful in the future. Then, okay. Conclusion and summary, let me go. I think very famous Microsoft database developer called Jim Gray, he proposed fourth paradigm. Fourth paradigm means first paradigm is experiment. Second paradigm is theory. Third paradigm is simulation. Then he proposed fourth paradigm, data-driven scientific discovery, which is now called data science. So sometimes fourth science, that's a data science. But I believe we have to go one more step. I may say fifth paradigm. This is data-driven scientific innovation. Not only discovery, we have to go to application. So from here, we can see clear vision in the future of genomic studies. Like if that can be applied for medical science, then we have to have like informatics, sequencing-based genomics, and industry. Those tripartite combination will be crucial to see the future society. Already we are having a genomic diagnosis, and moreover, such as Apple Watch or Fitbit, just like a watch type, and everything can be measured, not only heartbeat rate, but also like blood pressure. And those information can be utilized to maintain your health. So therefore, it will contribute to huge reduction of hospitalized patients. Moreover, we focused on marine metagenomics, but obviously we can have airborne metagenomics. Even when you are going to a supermarket, just you take air, then we can isolate DNAs. Amazingly, most of DNAs in any section of a supermarket, it's a human. We are producing a huge amount of DNAs from our own body, which would be utilized. So many cases, of course, health point of view, we have to consider. And not only human, soil, and the seawater, and the airborne. Anyway, I think so-called biome would be a very important features, particularly gut. Inside the gut, we have a huge community of microbes, which is deeply related to our diseases. So, for example, when you are interested in marine environment, as I said, that even you can construct the ship, then this ship would take sea samples, immediately sequencing can be done on the ship, then data information system, and which can be shared over the world. Then it will be a very, very important possible development. So all of those possible development, or now it's going on, I would call genome information society or genome information oriented society. So in genome, term genome was invented by Hans Winkler. Then Giorgio developed the structural evolution of genomics. Then we moved to innovation by utilizing genomic information. Those new society might be called Genome Information Society. So anyway, I think a DNA should be for world peace, and those are acknowledgement, and thank you so much for your attention.
Thank you very much. Chiamerei il collega Giovanni Caprara a presiedere un breve dibattito, ovviamente ringraziando tutti i nostri ospiti. Chiamerei qui anche il professor Danavaro.
Buongiorno, anche da parte mia. Ho seguito questa mattinata di incontri molto interessanti che hanno sollevato aspetti molto particolari ma importanti. Ringrazio il Presidente De Concini e il Presidente Danavaro per l'invito a coordinare questa fase conclusiva dell'importante incontro organizzato tra Napoli e Roma. Un'occasione molto importante, direi, per molti aspetti.
Good morning. I've been following these meetings, both which took place in Naples and here in Rome, and which were absolutely specific, but also very, very important. And I wish to also thank the organizers for giving me the opportunity to preside over the last important part of this meeting, which is going to be the question and answer session.
Ringrazio anche coloro che ci seguono in remoto, soprattutto i giovani attratti da queste importanti discussioni che hanno a che fare con la vita di oggi sul pianeta. È trascorso un secolo, appunto, come sappiamo, dal pronunciamento di questa parola, e questo secolo ha dimostrato come le ricerche sulla frontiera genomica siano, oltre che importanti, complesse, molto complesse.
I also wish to thank, in particular, all people who are connected remotely from home. And in particular, I wish to thank all the young people who may be connected and following this very interesting meeting. This meeting is about important themes for life, and especially in this century and all along this century, we have seen, because it has been demonstrated, that research in the field of genomics may have a very important impact on everyday life.
La genomica è anche una grande sfida della scienza e della tecnologia. Ricordiamo tutti, credo, negli anni 90, quando il professor Renato Dulbecco, premio Nobel, sosteneva alla guida di un ristretto gruppo di scienziati la necessità di realizzare una mappa dei geni degli esseri umani, ed era criticato perché ritenuta una sfida irraggiungibile, oltre che estremamente costosa, che avrebbe privato risorse ad altri campi di ricerca nell'ambito della salute. Ma si è visto che l'impiego massiccio della tecnologia, soprattutto informatica, a cui ha fatto ricorso prima di tutto Craig Venter, ha permesso molto più rapidamente del previsto la realizzazione della mappa genomica, che poi è diventata ovviamente uno strumento di base.
We all know that genomics is a challenge. It is a challenge for both technology and for science, a very complex one, by the way. And we all remember back in the 60s when Professor Dubecco, later on to become a Nobel laureate, actually said, and he used to preside a very small group of scientists and researchers, that there was a need to map the human genome. And for doing so, for saying so, he was fiercely criticized by some because such a goal was considered to be non-reachable and especially very, very expensive. And this would have certainly deprived and taken out some resources, I mean economic resources, from other important fields of human health. But actually we know that as the information technology progressed, these research studies and experiments could be accelerated so that the map of the human genome became a more affordable challenge as we know now.
Ma la mappa dei geni che dal 2000 è stata definita è solo un punto di partenza di una sfida molto più ampia della ricerca biologica che ha davanti diverse sfide, appunto, dalla proteomica all'epigenetica, e quindi pone delle domande che sono sempre più variegate e estremamente profonde dal punto di vista scientifico per trovare risposte. L'interazione con l'ambiente in particolare è un elemento molto importante.
So we know today that the mapping of genes in the year 2000 wasn't but a starting point. But now we have in front of us a much wider and much deeper challenge for science, for instance, biology, but also many other fields of science, such as proteomics, and from proteomics to genetics. And these are issues that appear to be more and more varied and deep and important to understand. Last but not least, for the environment's sake.
Ma io direi che questo aspetto della genomica rappresenta un capitolo sicuramente fondamentale di una tematica molto più ampia e anche molto più sfidante, se vogliamo, dedicata alla ricerca della vita in generale. Non sappiamo come la vita sia nata sul nostro pianeta, non sappiamo come possa evolvere anche in funzione dell'interazione con l'ambiente, nel quale noi abbiamo un peso sempre più rilevante. Non sappiamo quali strumenti molto spesso adottare per approfondire questa importante sfida della vita in generale. Vorrei ricordare in proposito un elemento che ha colpito particolarmente e che riguarda la ricerca della vita al di fuori della Terra, perché credo che il discorso sia appunto molto più generale. Noi cerchiamo la vita sulla Terra, la cerchiamo al di fuori, su altri pianeti, cerchiamo cioè le regole fondamentali di come la vita può nascere ed evolvere nell'universo.
I would say that this particular aspect of genomics is very, very important, but it is the whole chapter of wider and broader research about life in general, how life was born, how life has evolved over time, also in the picture and in the framework of interactions with humans. And we know that humans, especially on the environment, are having a very deep impact, and they had it in the past. And now we need perhaps new tools to try and solve this and resolve these challenges, let's say, and the main challenge being the searching for life on Earth and out of Earth. And this is one of the essential issues that we have to face today.
E a proposito di tecnologia dedicata alla ricerca della vita, mi piace ricordare una domanda che si sono posti gli esobiologi impegnati nella costruzione degli strumenti imbarcati sulle sonde Viking che nel 1976 sbarcarono su Marte con l'intento di cercare tracce di vita. Tracce che non hanno trovato, al di là di alcuni discussi segnali arrivati dalle analisi compiute in loco sul pianeta rosso. E quindi si sono posti una domanda: ma noi cerchiamo la vita con gli strumenti adeguati? Quindi la tecnologia oggi che cosa si deve inventare? Quale deve essere il processo di sviluppo tecnologico adeguato per cercare risposte che riguardano la nascita della vita, la sua evoluzione e l'utilizzo anche delle cognizioni che si raccolgono in questa ricerca perché possano essere utili a potenziali applicazioni future.
Talking about technology, especially in the framework of this search for life, I would like to remember those researchers who actually were looking for the most adequate tools to search for life, in particular during the Viking mission and the Viking probes back in March 1976, which actually gave no traces of life on the red planet. But for a few signals, however, which were actually considered questionable by some people from the red planet. So the question emerged. Are the tools we are currently used adequate for the challenge? And so what should technology do, and what tools should technology put at our disposal in order to answer to these basic questions having to do with the birth and the evolution of life, and also the use and the right application of such knowledge?
E quando parliamo di vita, ovviamente il discorso è molto più generale e molto più complesso, anche guardando la Terra, perché noi abbiamo realizzato la mappa dei geni degli esseri umani, la mappa, il genoma di alcuni altri esseri e altri animali, ma appunto noi dobbiamo ricordare che il pianeta Terra è per il 70% ricoperto dalle acque, quindi ci sono forme di vita molto più ampie e complesse da investigare. Sappiamo appunto quanto poco si faccia per approfondire le indagini oceaniche e della vita che nascondono, alle quali si aggiunge ovviamente la variegata forma di vita sulle terre emerse che riguardano dai vegetali agli umani e agli altri animali. Quindi il panorama della ricerca riguardante la vita tra la Terra e gli altri pianeti è una magnifica sfida che deve essere, a mio avviso, guardata in una maniera molto complessiva e generale, proprio per utilizzare tutte le possibili attività che possiamo affrontare in una maniera integrata fra loro, secondo anche una logica di ricerca scientifica tra diverse discipline che oggi è fondamentale per raggiungere risultati.
So talking about life and more generally speaking, also life on Earth, certainly, and we have done quite well so far about this because we have mapped the human genes or the human genome, and some animals' genome too has been mapped already. but a lot still has to be done about the life in the oceans. And we know that as much as 70% of our planet is made up by water. And so this makes the challenge more and more complex because we have to look for all forms of life. And again, not much has yet been done for the oceans. And which everything we learn about the life in the oceans should be then integrated and connected to what we know about the soil, so the land forms of life. And so I think that, however, the search for life on Earth and other planets is really a fascinating challenge because but everything has to be put together and united in a more integrated and, I would say, a multidisciplinary way.
Su questa frontiera, che ovviamente è molto ampia, oltre che molto complessa, gioca un elemento fondamentale, e cioè il ricorso alla tecnologia. Tecnologia che va dagli strumenti di indagine agli strumenti di valutazione e agli strumenti di disseminazione dell'informazione stessa. Quindi un mondo di tecnologie integrate che può coinvolgere anche il mondo industriale sempre più intensamente per innovare delle possibilità che naturalmente sono essenziali per arrivare a delle risposte anche scientifiche determinanti. La tecnologia oggi ha grandissime possibilità. Non possiamo immaginare, ad esempio, negli ultimi anni, grandi scoperte come il bosone di Higgs per la fisica oppure le onde gravitazionali se non avessimo messo a punto delle tecnologie così evolute che hanno permesso di compiere questo balzo e questa attività di ricerca di base che per esempio le onde gravitazionali, come sappiamo, era ovviamente in corso da quasi un secolo dopo il pronunciamento di Albert Einstein. Quindi la tecnologia gioca un ruolo fondamentale nella ricerca e gioca un ruolo che ha a che fare anche con la politica, perché quando parliamo di innovazione, di tecnologie, noi parliamo di un mondo industriale nel quale si possono vedere degli sviluppi economici orientati a questi settori di ricerca. E oggi la ricerca è sempre più condotta con una logica diversa rispetto al passato, soprattutto per quanto riguarda le ricerche onerose e di ampio respiro, dove spesso si uniscono investimenti privati a investimenti pubblici. Una chiave questa fondamentale per mettere in moto dei meccanismi politici fondamentali per garantire un'evoluzione della stessa ricerca di base.
However, this is a new frontier which is certainly wider and more complex. However, technology plays a crucial role in research in general, in science, in terms of investigation tools, experimentation and testing tools, and dissemination of findings from research. So the world is and has to be increasingly integrated in terms of technology, and thanks to technology, also the industrial world, because innovating in technology means also innovating in science. And we know that technology may offer great opportunities. For instance, if we think of physics and X bosons, or the latest discoveries about the gravitational waves that actually have been investigated ever since Einstein times. And so we're talking about almost 100 years. And but technology is also important and has a role to play for politics as well and industry, because there are continuously technological developments which have an impact on industrial growth and development. And we know that now the research approach sometimes is different, meaning that public and private institutions very often collaborate. And this is absolutely necessary also with having political decisions in mind.
E vorrei concludere mettendo l'accento su due aspetti di questa sfida che il convegno di 100 anni dedicati alle ricerche genomiche ha prodotto, e cioè due elementi che hanno a che fare con l'uscita dai laboratori delle informazioni, delle conoscenze, delle cognizioni che si conquistano nella ricerca, e cioè la informazione che ha a che fare con la disseminazione dei risultati e con la comunicazione. La disseminazione dei risultati e anche la comunicazione è strettamente legata alla tecnologia, come sappiamo, e questo è un elemento fondamentale per cercare di trasmettere non solo i risultati raggiunti, ma la loro importanza e le conseguenze che possono innescare nella società. E questo aspetto, che ha a che fare sia con strumenti adeguati di disseminazione tecnologica, come il professor Goggiabori aveva evidenziato nella sua presentazione, ma ha a che fare anche con una cognizione stessa dei significati, che è un elemento molto importante che si collega direttamente alla comunicazione. La comunicazione è un aspetto oggi determinante su qualsiasi frontiera di attività umana, soprattutto ovviamente per quanto riguarda la ricerca scientifica, perché deve seguire dei canali, dei metodi, degli approcci sicuramente molto diversi da quelli che si adottano nel mondo della ricerca in quanto tale per disseminare gli stessi risultati. Quindi è una doppia sfida quella della disseminazione e della comunicazione, che deve, a mio avviso, procedere in maniera molto stretta, e ciascuno deve inventarsi i modi adeguati per portare all'esterno questi risultati che si raggiungono. Anche perché abbiamo visto, proprio parlando di salute, una parola evocata molto spesso nelle relazioni che abbiamo ascoltato, il discorso della salute ha dimostrato anche nell'ultimo anno, purtroppo, o grazie, se vogliamo, alla pandemia, una debolezza nella cognizione da parte della società dei problemi che abbiamo davanti per quanto riguarda l'informazione scientifica. E questo significa che dobbiamo agire in maniera molto più profonda, cercando delle vie adeguate per evitare anche quei rischi di una falsa informazione che molto spesso può nascere dallo stesso mondo della ricerca, che se non è ben veicolato sia con la disseminazione che con la comunicazione può portare a risultati estremamente negativi per la società. Conosciamo tutti i fenomeni dei Novax, che non è solo un aspetto che riguarda il nostro paese, ma qualsiasi nazione, proprio perché sono generati da processi cognitivi di informazione e di comunicazione non appropriati. Quindi lo scienziato oggi ha una doppia sfida, parlando di genomica, parlando di ricerca della vita, che è appunto quella di comunicare in maniera appropriata le proprie attività, le proprie ricerche, nelle quali impegnarsi, credo, nella stessa maniera, con lo stesso rigore e con la ricerca dei metodi più adeguati per trasmettere questi passi fondamentali della ricerca, che ha a che fare con l'esistenza dell'uomo, con la tutela della sua salute che può derivare da queste ricerche e in generale con la ricerca della vita e la ricerca dell'evoluzione dell'uomo su questo pianeta.
in order to conclude, I would like to underline two main aspects that have been particularly emphasized in this meeting celebrating the 100 years of genomics. And the first one has to do with the way in which information and findings and results from science and scientific experiments and so on get out of laboratories, meaning that the way in which information is disseminated and communicated to the general public. And this is, of course, linked to the availability of adequate technology to do so, because we know that the way in which information is spread has an impact on the society as a whole. As Dr. Gojabori has rightly said, this is very, very important. And certainly communication is important for all aspects of our life, and not only for science, of course, and therefore methods, new methods and new approaches might or should be identified, because they are different from the methods that are being used in laboratories to communicate and disseminate. So I have to say that scientists in this respect have a twofold challenge. Of course, first of all, they have to be rigorous and accurate and whatever and competent in science, but they must also learn and develop a correct way to spread the information out of their labs. And for instance, let us take the example of public health. Recently, we have seen how important the dissemination and the explanation of knowledge to the general public is important. And today, due or probably sometimes thanks to the pandemic, we have identified this point of weakness. The point of weakness is very often the lack of awareness in the society when it comes to science. So this is a risk. This is a risk that can and must be prevented, but we all have a role to play in this. And because we have to fight fake news, for instance, and even communication and information spread by scientists may cause confusion, and because it has to be communicated in the correct way. And of course, this is very, very important for the continuation, I would say, of life, of our life. And therefore, the communication should be appropriate. And I think that so rigor should be applied, and rigor should be applied, and accuracy should be applied in the communication on the part of scientists and researchers. And this is for the sake of life and human life in general.
Grazie, e a questo punto credo che le domande possono essere poste qui e anche tra coloro che ci seguono.
Thank you so much. Now questions?
I would only say that we should be rather quick because we have to go for lunch and so on. But please come. Or maybe you can.
Grazie, io avrei qualcosa da dire proprio sul rapporto tra scienza, conoscenza, tecnologia e poi la diffusione e quindi la comunicazione. Io non so se sono riuscita a cogliere l'obiettivo del mio discorso. Io ho partecipato in maniera veramente importante a una scoperta di base e adesso mi tocca sentire a vari livelli che nessuno sa che cos'è. Allora, cominciamo a chiarire quali sono i concetti della vita. Cominciamo a chiarire che cos'è la tecnologia. Io, per esempio, sono stata 10 anni mandata dalla delegata della Comunità Europea alle 3 prime banche dati, quella dell'IML, quella della Genome, cioè di bioinformatica, anche fino a questo punto, e quella del DDBJ, e adesso noi comunichiamo delle cose in maniera del tutto, secondo me, aleatoria. Quindi mi permetto di dire che la scienza deve essere portata più avanti e non è tecnologia. E la tecnologia come è attuata. Vogliamo fare i followers, vogliamo comprare la macchina ultima Nespresso perché socialmente i follower, quella pizza non mi ricordo l'ha fatta. Quindi io sono, a prescindere da tutto, io vorrei sapere quanti di italiani sanno che la fecondazione artificiale e che il processo di fecondazione in che cosa consiste. Non sono in disaccordo con le novità, intendiamoci, ma lei sa bene che abbiamo delle leggi in Parlamento, cominciando dall'omofobia. Io non sono niente affatto contro gli omosessuali, intendiamo, ma la riproduzione è fatta in una maniera e questo, e quindi è quello che fa acqua, è la formazione. Mi scusi.
I would like to... Sorry. I would like to say a few things about science, knowledge, and technology, and of course about the dissemination and the communication of the content. I participated in a very important discovery, but I now found out that nobody knows about it. And therefore, I have taken part and I have contributed for 10 years in 3 very important data banks at an international level. But I have now observed that we communicate the results of these efforts in a very vague and inconsistent way. So science should be carried on. It isn't only a question or a matter of technology, because, okay, we want to be followers, we want to use the espresso coffee machine, okay, but there is more to it. And I don't know how many Italians know what IVF, what the in vitro fertilization is. And we know there are so many laws about reproduction being discussed in the parliament, but I agree, everything. I have nothing to do with, I'm not against homosexuality, of course, but I think that a greater effort should be done in terms of basic science and the results that we achieve in the field of basic science.
Okay. So if there are no other questions, I would like to ask Roberto to close, and then we have the award of the prizes, and then we
so just a few words for concluding. A number of issues, and I'm hoping that during the dinner and in the follow-up of the meeting we can also exchange some ideas. At the end of these 2 days of the meeting for 100 years of Genome and the perspective for human and planet life, I could say that life is under threat. I mean, we are jeopardizing all forms of life on Earth. We are jeopardizing human life. The COVID experience is demonstrating how weak we are. And this Congress demonstrated how much we are still far away from getting the basic information. The sequencing, the genome sequencing is just having the basic information without even understanding the functions, the potential we have. So a few excellent ideas have been proposed. and among them, two main sectors, I would say. From one side, understanding better, not just from the human genome, what we can learn, but also from the interaction between the human genome and the environment. Understanding, thanks to the exposome, that our life is threatened by the conditions, by poverty, by education, by diet, lifestyles, and whatever. And this is a matter of equality. From the other side, we were also attracted by searching life in the exoplanets or whatever. But there's a kind of tale, because we have on Earth from 2 to 10 million species. And we have made the genome of just a few dozen of them. So we have had million species to sequence. And we are looking for the life in extraterrestrial. This is part of the communication. This is part of the power of certain fields of science that are more attractive than others, and that probably they also touch better our fantasy, but they do not really explain that our life is tightly linked to small pieces of genomes that are viruses, to the creatures around us that we're missing, losing. We have destroyed 75% of the terrestrial biomes, altered 2/3 of the blue planet, and we are fighting against very minuscule organisms and sometimes losing. So now we have a big challenge in front of us. And the conclusion of this meeting and the proposal by Professor Gojobori had is to go ahead, to go for the global genome, for the genome of the planet, maybe starting from the sea. And the marine metagenomics it was showing today, where the most of life is hidden and the majority of genes is still present and undiscovered. The hope for all of us is that this is just giving some ideas, some inspiration for the young scientists and for all of us to understand that maybe if we want really to achieve these important objectives, we need much more than a few scientists around the table like we are. We need to look for an engagement of the governments. We need for a global coalition of scientists, because the challenge we have had is huge, and the time is short, and we need to do much more for sequencing the life around us and for understanding the secret of life, including human life. So thank you very much for everything. I give the floor to the President Concini for the awards of the young scientists, and we look forward to see you next at the restaurant.
So I would like to add one more. I think the whole subject needs also some theoretical approach. I'm a very theoretical scientist. In fact, I'm not even a scientist. I'm a mathematician. And I think that the challenges posed by Takashi Koshibori to understand how to organize is a challenge which has to do with technology, but also with introducing some ideas of symmetry, of which are both Galilean or mathematical in spirit. And I think this is an enormous challenge, and it could simplify the whole situation, because my impression is that at the moment the situation is too complicated, and if you have too much information, you tend to have no information, as you all know. So this is my, being out of this subject, my advice for the future to the young scientists. Now we have two very brilliant young scientists, and so I invite them to the floor, and I don't know. Is there a check or something, or no? So it's a virtual check. Later. But so the first winner is Umberto Palladini, and he has been noted for the interesting results on the improved reference genome of the herbivoral vector Aedes albopictus. So I mean, solid thing will come. But for the moment, thanks to Davide Fasolo and Paola Bonfante, who gave us permission, we want to present this diatomy picture, which is I think very nice. It was done by Davide Fasolo for the other prize, for the Fasolo prize. Thank you so much. You are from Pavia, right?
Yes.
The second winner is Miriam Rocco, who is from Naples, from Dorna. And she has been noticed for the interesting results reporting on the molecular signatures of clonal integration in Posidonia oceanica live storage. And I want to thank you, especially because I learned a lot of Posidonia to understand you. So again, we have this picture. Okay, so let's congratulate. So now I declare the meeting over. You are free to run away. But if you want to join, there is a lunch in the restaurant Limonaia, which is just in front of you. Okay.